Hello viewers, welcome once again to our ongoing series on Mendelism and beyond. In 2 or 3 of my last lectures, I had given you a very detailed account of linkage, crossing over, and of course the cytological basis of crossing over. If the viewers remember correctly, there were some very classical experiments which were performed by Professor T.H. Morgan himself, followed by those of Crichton and Barbara McClintock on Zea mays, and then later by Kurt Stern on Drosophila, which proved beyond doubt that yes, there was a cytological basis for crossing over. We had also talked about linkage and how the linkage groups existed and what was their relationship with the chromosome number or the diploid number of bivalents. In this whole discussion, we came to a conclusion that recombination frequency was a very important parameter. Today, viewers, let us pick up a thread from our last lecture on recombination frequency, and let us see today how we use this particular parameter in constructing the linkage maps. Our earlier discussion, as we have seen, has highlighted some very important points. The first being, this is for our recapitulation, that the number of genes are usually much greater than the number of chromosomes. This would obviously mean that many genes would reside on the same chromosome, and if this is so, the genes which are linearly arranged on the chromosome, they would all tend to get inherited together as a single unit, that is a single linkage group during meiosis. Another aspect we should not forget is that the number of linkage groups would be equal to the number of homologous pairs of chromosomes or the number of bivalents of a particular species. You remember, learners, we had talked about the exceptional nature of the number of linkage groups in human beings, in the males as well as in the females. We reached a conclusion that in case of the human female, it would be 23, and in case of human males, it would be 23 plus 1 equal to 24. And then another point would be that these linked genes, they are usually staying together. If they are usually staying together, when would they separate? How would the recombination occur? Obviously, crossing over has to take place, but then we have to now analyze where and with how much frequency would the crossing over take place. But one thing is very clear, that closely linked genes would have definitely less chances of crossing over than those which are widely located. And lastly, each gene definitely has an order of location on a linkage group. So that means whenever we are going to talk about crossover frequency, it seems to be constant for two closely linked genes, and it is also intimately related to the physical distance between the two genes as they reside or as they are located on a chromosome. In other words, we are going to adopt crossing over as a tool, and by using this, we can find out the exact distance between the two linked genes and what is their order of location. This parameter is going to help us to construct what we call as a linkage map. It is also popularly called as a genetic map, and if our study is based on crossover, naturally we can also call it as a crossover map. Before we go to the details of recombination frequency, viewers, on your screens you can just recapitulate how crossing over was being operational at the DNA level. On your screens you can find that the DNA molecule, which is of course double stranded, would have nicks or breaks at certain points, and these nicks would now be subjected to strand invasion, and of course it would be later assisted by base pairing. Once the base pairing or crossing over had taken place, then comes the time of repairing. So it was a cut and paste phenomenon, as we had discussed in some of our earlier lectures. So after ligation, now recombinant chromosomes would come into existence. Let us then see how the frequency of recombination can be used as a very effective measure for finding out the degree of linkage. One thing is very clear: any pair of alleles you should know, and the gametes which are going to be produced through the process of meiosis, there can be two situations: parental combinations and the recombinants. Now they are definitely going to be based on whether it is a case of coupling, that is cis, or repulsion, that is trans. We have had a very detailed discussion on this aspect. Just to give you again a brief of the parental alleles, if they are present in coupling or cis configuration, after meiosis we can have parental as well as the recombinants, and so would be the case if the parental alleles, they are present in a trans configuration or in a repulsion phase. Now Professor Morgan performed various interesting experiments to get to this whole idea of how to create a map, or at least how to quantify the distance, the order of the genes as they were located on the chromosome. So he detected mutations in the X chromosome of Drosophila, and he used this idea for the study of linkage. Learners, let me remind you, in case of male Drosophila, we find that there is the Y chromosome which is showing total linkage. So this entire story is revolving around the X chromosomes. So he took the characters as W plus, which was the wild one, as the normal red eye color, and obviously then the recessive W was white eyes, and then the wild, the dominant one, was normal wing size, and the miniature wing size was represented by small m. Now in this particular cross, you would find on the top that we have mentioned W plus M by Y, and not a pair of alleles, because you know that the male is going to be a hemizygous case. There is only one X chromosome in a male, like we have in human beings. So when this white-eyed females were crossed with miniature wing males, the mutant alleles in the homologous chromosomes, they got together, and in the F2 generation, the progeny that we got were parental types. They were actually calculated to be 66.5. How did we arrive at this percentage? As you can see on your screens, 226 plus 202 divided by 644, and these were the parental combinations. The recombinant types were only 33.5, that is of the non-parental allele combinations. So this was 114 plus 102 by 644. Now this result is definitely at variance, at a lot of departure from the expected 1:1:1 ratio, which happens to be the test cross ratio. So there is no question of independent assortment here. We find that the genes are no doubt present on the same chromosome, but they are not showing 100% linkage. If there was complete linkage, then there would not have been any question of the origin of the recombinants, and non-parental combinations should have been 0. But then in fact it is 66 versus 33. That means this is the frequency of recombination, that is the 33.5% of the recombinants. This suggests that out of 100, 33.5% are the non-parental combinations. This is definitely in contrast to the 50% recombination that we normally expect, although we find that if the genes are very far apart on the same chromosomes, again they will have a crossover in between and they will show independent assortment. Morgan was not very sure as to what the phenomena would lead to, so therefore he performed another cross to cross check his ideas. Now in this case you can find that this cross would now be having white-eyed and miniature wing females to be crossed with the wild type of males. And in this cross, he again came to a conclusion that the recombinants were around 37.7. You remember that we were talking about 35.1 in the last cross. So it is quite near that particular figure. And the parental types were 62.3. So this also happens to be quite near the 65% mark. So that gave a very, very clear idea that the frequency of recombination is the same if we are talking about a coupling situation or a repulsion process, that is of the heterozygotes. So it really doesn't matter how the alleles are arranged, the frequency seems to be similar. So how do we now use this recombination frequency to our advantage to quantify or to construct a map? So we usually represent this linkage of genes in the form of a genetic map or a linkage map, as I said before, or a chromosomal map. This was developed by Alfred H. Sturtevant in 1913. He was, I have repeatedly said that he was one of the most illustrious students of Professor Morgan. So actually a genetic map that he made shows a linear order, and it is so spaced on a chromosome that the distance between the adjacent genes is proportional to the frequency of recombination between them. So that means there is a relationship between the frequency of recombination and the distance which is there between the two adjacent alleles. Just to give you an example, if two genes are recombined with a frequency of say 3.1%, how far they would be on a chromosome? Sturtevant said that in that case they would now be 3.1 map units apart. He said that one map unit is going to be a designation for 1 centimorgan. Centimorgan is a unit which was given by Sturtevant in honor of his teacher Morgan. So we could use either of these units. 3.1 map unit is equivalent to 3.1 centimorgan is equal to 3.1 frequency of recombination. Learners, you must remember 1 centimorgan would be equal to 1% recombination frequency, and which means 1 map unit. If we talk of human beings, how do we apply this centimorgan unit? We say that 1 centimorgan, which is going to designate the distance between the two adjacent genes, would be roughly equal to say 1 into 10 to the power 6 base pairs. In fact, 1 centimorgan would equal 1 megabase, which carries around 15 genes. The logic behind this calculation seems to be that our genome has about say 300 megabases, and they contain about 45,000 genes. So keeping this whole idea into account, another cross you can see on your screens is between the white-eyed females and the diminutive, that means the size is less, of the males in Drosophila. It is just to signify the point that how the recombination frequency can be expressed in terms of the distances. Now, in the F1 generation, we get a trans heterozygote, and in the progeny, we find that the parental types happen to be 969 out of 1,000, which means that 96.9% have not segregated at all, and they wanted to remain together, and it was just the remaining part which was the recombinant part. So, the recombinant part, if we add up, comes to 31 out of a total of 1,000 progeny. So, the recombinant types would be amounting to 3.1%. How do we express this in different languages? Genetic distance would mean frequency of recombination, which means 3.1% is equivalent to 0.031. And percentage recombination is of course 3.1. What is the map distance between the two adjacent genes? It would be 3.1 map units, which is then equivalent to 3.1 centimorgans, written as 3.1 small c capital M. And on a scale, on your screens you could find, we could represent this graphically by having the diminutive size and the white eyes at a distance of 3.1 centimorgans. Let us take another example. We are talking about 50 meiotic divisions. Now, this 50 meiotic divisions obviously would give rise to how many haploid cells? 50 into 4, which is 200. And we observe that out of these 50 meiosis, 49 meiosis do not show any crossover. So, if they are not showing any crossover, then in 49 into 4 cases, which happens to be 196, we get non-recombinants, that means the parental ones. And one meiosis would give rise to 4 haploid cells. Out of these 4 gametes, 2 are non-recombinants, which we add in 196, it becomes 198, and just 2 happen to be recombinants. Learners, you must remember that we are talking about 50 meiosis, 2 recombinants. This is how the calculations would move further. So, what is the frequency of recombination? The frequency of recombination R would mean 1 recombinant plus 1 recombinant, that is 2, over a total of 200, and this happens to be 1%. So, that means these two genes are 1 map unit or 1 centimorgan apart. Now, here comes the catch. 1% recombination means that one meiotic cell in 50 has a crossover in the region between genes, and if one of the 50 myocytes has a crossover, then the frequency would be 1 by 50, which is equal to 2%. Now, crossover value is 2%. And recombination frequency is 1%. Now, this seems to be a little confusing. Let us see. The frequency between the genes is 1%, as the crossing over is resulting in 2 recombinants and 2 non-recombinants, but crossover frequency is 2%, because we have just mentioned that out of 200 chromosomes, due to 50 meiosis, only 2 chromosomes, in other words 4 chromatids, are the recombinants. So, the logic goes that as only half of the chromatids in each cell with a crossover are actually recombinant, 2% crossing over in fact corresponds to 1% recombination. Let us take another example to prove our point. In the cross, which you can see on your screens, we are talking about a test cross between small a, small b, small b, and crossing it with capital A and small b. Now, we find that below we have given the figures of 135, 430, 390 and 120 of different individuals. Now, within this cross, if you see carefully, how do we get the recombination frequency between A and B? it would be, we are taking the 135 and 120 together over total number of individuals to be 24%. In other words, just to give you another example so that a learner may get the whole idea seeped into his minds as to how to juggle with the recombination frequency and the crossover frequency. If a F1 hybrid having a genotype capital A small b and small a capital B produces 8% crossover gametes, and these crossover gametes are AB and AB, then what is the distance? The distance between A and B would be estimated to be 16 centimorgans or 16 map units. On the other hand, if we take another example, if the map distance between the gene loci capital B and capital C is 12 centimorgans, then it means that 12% of the gametes of the genotypes capital B oblique small b small c should be of the crossover types. That means two types are there. So that means 6% of capital B small c and 6% of small b small c. I hope I have proved the point. And each chiasma, on the other hand, produces only 50% of the crossover products, and 50% crossover would therefore be equivalent to 50 map units or 50 centimorgans. So if now we know that the average or the mean number of chiasma are known to us for a chromosome, how can we find out the total length of the map? Because we have to have a limit to that map also. So the total length of the map in that case for that particular linkage group would be equal to the mean number of chiasmata into 50, which would now indicate that we have to set the limits also. So this is a very preliminary account of the linkage maps. We are yet to talk about how the double crosses, the triple crosses, and of course what is the order of genes on the linkage map, and how the linkage maps are being now made on model lines at the molecular level. We will talk about these aspects in some of our forthcoming lectures. Thank you.